So to get started, we will begin with a short introduction that will be given by my colleague, Dr. Will Seiberts. He is in scientific sales. He's coming from the University of Göttingen, and he has a strong background in cardiac electrophysiology, particularly in atrial fibrillation. Without further ado, I think we can start. And whenever you're ready, Will, you can start sharing your screen.
Great. Thank you very much, Jema. Hopefully you can all see my screen at the moment.
Yes.
Thank you for that introduction. Yes, I thought it would be pertinent to give a little bit of a brief overview about the technique of automated patch clamp before we get into the main talk today. Automated patch clamp is, of course, very relevant for the study of cellular electrophysiology, but it is patch clamp in a way that may not be as familiar to many in the audience today. So I'd like to give a brief overview of how this technique is different to the conventional patch clamp that we usually use in the lab. Now, many of us will be familiar with conventional patch clamp, which involves the manual micro-manipulation of a very fine glass pipette onto the cell of interest. And seen in this cartoon here, you can see this pipette slowly approaching the cell. It forms a stable seal against the membrane and eventually ruptures that small patch of membrane at the tip of the pipette, which gives us electrical access into that cell. This is then, we're able to use this to then quantify the global activity of all ion channels residing in the cellular membrane. Now, this is the gold standard for studying ion channel activity and has been that way since the 1970s. It does come with a few disadvantages, however, and that is that it is a very low throughput method. Cells can only be measured one by one using this technique. It is also very time consuming, a very complicated technique that involves significant training and technical competence. It involves a lot of bulky and expensive equipment involving inverted microscopes, large Faraday cages, anti-vibration tables, and the necessity to pull and fire your own pipettes. So it does come with these disadvantages as well. Planar patch clamp is what we're going to be talking about today. And this is patch clamp without pipettes, essentially. Now, how does this actually work? Planar patch clamp relies in very, very small holes penetrating through a borosilicate glass substrate. Now, if you imagine that we place cells in suspension onto this substrate, a cell will be attracted to this hole through the application of negative suction pressure. A stable seal will be formed in the realm of a gigaohm, and then continuous suction pressure will eventually open this membrane, giving us electrical access into the cytoplasm. And here you can see this electrical configuration is analogous to that of conventional patch clamp. However, we've removed the necessity for that experimenter dependence. We do not need to be there and try and approach the cell with our pipette. What this also means is that we can basically get rid of a lot of this complicated, bulky equipment as well. We no longer need to visualize what we're doing. Therefore, we don't need inverted microscopes. We don't need anti-vibration tables or large Faraday cages. Instead, the small recording chamber can be electrically isolated to prevent noise. And we also don't need to pull and fire our own pipettes either. All we need in the end is a small recording chamber that can sit on our desktop, an associated amplifier, and some kind of PC or monitor for us to be able to visualize our results in real time. So a much more streamlined approach to the patch clamp technique overall. There are several recording configurations that we're able to use using this technique. The first here is the on-cell or cell-attached configuration, where once a cell is attracted to this patch clamp hole, the membrane is not ruptured. And that means that all ion channels that reside within that small patch of membrane over the hole can be measured as they open or close. And in this representative trace here, we can see the opening and closing of 1, 2, and 3 ion channels as they open and close consecutively. If we apply suction to this configuration, we rupture that patch of membrane, and therefore we're in whole cell configuration. And this therefore allows us access to measure the entirety of the ionic activity of all ion channels residing within the cell membrane. The application of a stimulus, such as a change in voltage, a change in heat or mechanical pressure, or the application of a ligand, or even the application of light, if we're using optogenetic constructs, will allow these ion channels to open and ions to flow in or out of the cell on a global scale. And then we're able to measure the flux of these ions in the form of this trace shown here. Once the stimulus is ended, we can therefore also measure the desensitization or the inactivation of these channels as well. Now, a third configuration that's not shown here in cartoon form is perforated patch. And this involves the application of a perforating agent, such as an antibiotic, into the internal solution, which penetrates the membrane with very small pores over that patch clamp hole. And this keeps the cytoplasm intact, but it still allows us electrical access to the interior of the cell. And this can be useful for some experimental applications. Now, at Nanion Technologies, we've incorporated this planar patch clamp technique into a variety of instruments. And I'd like to give a little bit of an overview here of the different instruments that we offer. The first is the PortaPatch Mini here. Now, this is a very compact patch clamp rig, which is able to measure one cell at a time, and is usually used for basic tests of cells and ion channels. It is the most compact and the smallest patch clamp rig available on the market today. The standard PortaPatch model can also patch one cell at one time, but it involves the use of various add-ons for continuous perfusion of external and internal solutions, as well as sensitive temperature control and the application of optogenetic stimulation as well if required. Moving up into the throughput or the more higher throughput capable machines, we have the patch liner here. This is able to measure 8 cells simultaneously at one time. It contains a robotic liquid handler, which means that there is no manual need to deal with solutions. It can all be done automatically. The use of multiple channels at one time increases the throughput dramatically. And this essentially means that we're able to acquire a lot more data from a lot more cells within a given experimental window. It's a very versatile and flexible patch clamp device, and also contains add-ons for precise temperature control and dynamic clamp for the measurement of action potentials from excitable cells. Now, our hallmark device, the SynchroPatch 384, is our high-throughput automated patch clamp device, which is able to measure up to 384 cells simultaneously. As you can imagine, this means that we're able to acquire a huge amount of data within a small experimental time window. It contains precise temperature control and current clamp as standard, and can also be used in so-called 32 well mode. This is for when the use of all 384 channels might not be necessary for a given experiment, and one can customize how many channels one wants to use at a given time. Now, we'll be hearing a lot about this device in our talk today from Dr. Chang Ng, and I'd like to give a little bit of a snapshot into how exactly this device works, because a lot of the magic actually happens within the 384 well recording plate. Now, this might look like a typical cell culture plate on the surface, but what's not shown here in this cartoon is the base of this plate is actually a very thin borosilicate glass substrate. And within each of these wells is located that patch clamp hole. Here's a little cartoon of what an individual well looks like with that hole in the middle. And once cells are automatically deposited via robotic liquid handler in solution, the application of negative suction pressure from underneath will attract a cell onto that hole. A stable seal will be formed, and then the experiment can continue. This represents an extremely powerful evolution of the patch clamp technique, and will be the base of our talk today, where Dr. Chang Ng will be using automated patch clamp, or APC, for enhanced variant assessment in cardiac channelopathies. Now, I'd like to give a brief introduction to our speaker today. Dr. Chang Ng, he leads the ion channel functional genomics team in cardiac electrophysiology at the Victor Chang Cardiac Research Institute. His impressive list of achievements include developing the first high-throughput patch clamp assay for KCNH2 that satisfied the recommendations, which were published by the ClinGen Sequence Variant Interpretation Working Group. He has formed a strong collaborative network in the clinical diagnostics industry, connecting academia and industry partners from Australia, the US, and the UK to implement his high-throughput patch clamp assays and improve genetic diagnosis capabilities for rare diseases. Cheyenne, we're really, really thrilled to have you here today. Thank you very, very much for sharing your work with us. Without further ado, I'm going to stop sharing my screen, and then the floor is yours.
Thanks, Will. Thanks for the very detailed introductions because I'm not going to talk about all that. I'm just purely going to focus on genetics and patch clamp on cardiac ion channel. So it's very nice that you describe that to our audience. Before I start, I thought I'm going to do a quick poll to survey what we have today, different professions in our audience. So I'm going to do it myself. So you just scan the QR code and then select one of the options that best describe your profession. So we can all see who's in the audience. Okay. So we have about 10 or close to 15% of healthcare professionals, some students, and as expected, mostly are science and research academic. All right. So let's move on to my next slide. I'll put my pointer on, laser pointer. So today I'm going to talk to you about how I use the patch clamp to enhance variant assessment, as well as some risk predictions and penetrance determinations in cardiac channelopathies. A bit of background. So I'm based in Sydney at the Victor Chang. I'm from Malaysia originally. And some of you might not know about Victor Chang Cardiac Research Institute. So it was named after Dr. Victor Chang. He founded the National Heart Transplant Program at St. Vincent Hospital. And between 84 and 91, he had transplanted more than 197 heart transplantations. But tragically, he was ill in 1991. And I think about 3 years later, Victor Chang Cardiac Institute was established to honor him and name after him. So the topics that I'll start off with is sudden cardiac arrest. So sometimes you will see the news of runners collapse. So for example, this example is 2014. Is 11 years ago now. Runner collapse near the finishing line in a city to serve marathon event in Sydney, and you get to see this if it's reported in the news, but sometimes it doesn't. So last year there were 10 cases of syncope and 3 cases of sudden cardiac arrest, but this wasn't reported in the news. I mean, the doctors that helping with the events and look after the patients told me the statistic. And I guess this is more prominent when the sports get prominent, and of course you'll get reported more often, as in soccer or football. Players, if they collapse in the field, you will know it the next day or immediately afterwards. So according to Professor Sanjan Sharma, about 600 young people under the age of 35 each year have a arrest and fatality if there's no defibrillator around or is undocumented as no one's around to perform CPR, then the chance of you surviving if the arrhythmias didn't terminate spontaneously, it is very low. But you might think cardiac arrest only happens if you do exercise, but that is not the case, because sudden cardiac arrest can also happen at rest. So in this case, Victoria just finished off one night typing some email, and she stands up and then she collapsed on the floor. And fortunately, her husband found her and performed CPR for very long time, I think 16 minutes, until paramedics arrive and give her a shot using the defibrillator. And she survived and well because CPR was performed properly because her husband is a lifeguard. And so in the case of you have a syncope or cardiac arrest and require medical attention, clinical assessment might be diagnosed you to have a long QT syndrome, in this case for Victoria, that likely to explain why she had an episode of sudden cardiac arrest. And what happens if you have a clinical diagnosis of long QT syndromes or any other inherited syndromes, in this case, anemia syndromes, but it can be any other inherited diseases? Then you'll be offered genetic testing through genetic counseling first, and then offer you genetic testing to identify, the genes that are known to cause your disease and whether, you will have a known disease-causing variants or not, because that will be helpful later. I can explain it a bit more. But these are some of the testing services that I collaborated with in Australia as well as overseas. But how do these clinical genetic testing services classify genetic variants that they identify through sequencing? They're largely based on the 2015 ACMG AMP variant classification framework. And it is basically a criteria for pathogenicity, which is about 16 here, and each code represent a different parameters, so to speak, or criteria. For example, I'll be talking about this code PS3, which is a functional studies. And if you're genetic counselors or clinical geneticists, you'll be familiar with this. But if you're not, these are just, random codes for you, I suppose. And the other one, I guess people sometimes will know is the PP3, the in silico prediction. So I guess, this one you might know a bit more if you do some research on predictions of the amino acid changes and what is the deleterious effect by alpha missense, for example, the latest one produced by Google. And in addition, there are also a separate list of criteria for benign, for classifying variant as benign, but they're largely, the stronger ones, it would be allele frequency, because if your allele frequency is more than the disease prevalence, then it's unlikely that variant will be contribute to the disease in that patient. I'm not going to go through all these things, but just simply to say the original code is using a combination of different, criteria. If it's contrasting or conflicting between the two, it'll be a VUS. But the easiest way these days to look at this is point system, as in if the S here means a strong, so you get 4 points, and the moderate, M stands for moderate, you get 2 points. P here in the middle is a supporting, you get 1 point. And then same with the benign one, you get a minus point. So if you do a point system here, you can have, all the different criteria come together, but to reach a likely pathogenic, you need a +6 points, and VUS will be 0 to 5. And as I said, I'm going to talk about PS3 here, which, in theory would be up to 4 points. But I guess we have to talk about the VUS first. And what is the VUS if you're not familiar with VUS? VUS stands for variance of uncertain significance. It is one of the 5 outcomes after genetic testing. So you could have, a pathogenic, likely pathogenic, benign or likely benign. And if unfortunately, if you get a VUS, then I'll explain later why, what VUS cannot do, I suppose. So the benign, likely benign, if it is common variant or, this day common variant don't even get filtered, they will be filtered out, so they don't even see it, I suppose, in some testing lab. And, the synonymous variant would be those, if not affecting the splicing. But this plot is trying to say here, over the years, the other classification doesn't really change much, but the VUS is going up. This is in the thousands, so you have like 200,000 variants here. But it is gonna more than that now. I think I don't have the 2025 stats on that. But I guess the first explanation to why would be because of more genetic tests were performed. So the more you do, and the more VUS you're going to find, I suppose, because you look for it in a way, because the more tests you do. And is also because of insufficient evidence that used by, the ACMG AMP criteria, which all these labs are using with a slight modification. And also, if you're dealing with rare diseases, that is also one of the reasons, more of them are VUS, especially if you have a missense variant in channelopathies, most of them will be VUS. And, in some instance, not applying certain evidence by the testing laboratory contribute to increasing number of VUS as well. So if you have a VUS result, it's medically unactionable, meaning you can be used it for clinical decision making. So the first thing is VUS is not a diagnostic test result, meaning you can confirm or deny, I suppose, what the variant is after the testing, which is sort of defeat the purpose of doing a genetic test at the first place, because you want to do the test and you identify a gene that known to cause your condition, and a variant hopefully is a likely path. Then you can, follow up with other, for example, family management. You can then have a genetic cascade testing to identify a risk family member, or family, pregnancy planning, IVF, and etc. And the other aspect, I suppose, sometimes we don't talk about it, is, you provide an answer to the proband, the one that had the genetic test, to be whether it's the cause or it's not the cause. Also to the family as well, because then you can do cascade testing to identify, sometimes the parents pass the gene down to the children. So, those kind of things are not, I suppose, people don't think about it if you're not dealing with genetic counseling. And sometimes in some certain diseases, you can also provide, prognosis, as in if it's a gain of function variant, you confirm that, and if the clinical phenotype is not very strong, such as in epilepsy area, you could then prescribe medication more effectively, I suppose. So this is all what VUS cannot do. So, which is why we, ClinGen, I suppose, trying to tackle this problem as well in a way, by promoting consistent classification, by establishing criteria specification for the ACMG criteria code. Some of them are no longer applied, like, BP6, I think, but these are just the code that people can use. And I'm a member of these two VSEP. One is the potassium channel, the other one is the sodium and calcium channel. So this would be largely cover all cardiac channelopathies space. And the other thing the VSEP hopefully will do eventually is also, because in ClinVar, sometimes you will get this conflicting classification by different submitter, and VSEP will have the capability in the future to review those conflicting classifications and, recommend the pathogenicity, I suppose, for that variant on the top high-level summary. So let's get into cardiac ion channels. So our heart rhythm is regulated by different ion channels. And this is just to illustrate that, what we have here is I means current, amplitude is a number of available channels multiplied by the probability of opening and the conductance. So this gives you the output, such as what we measure here for the sodium current. This amplitude will represent the function. And if you have a pathogenic variant that, in this case, cause loss of function, such as, you don't have signals anymore or current anymore in this sodium current or this potassium current here, this is wild type, and this is nothing, which is little current that you can measure, which is severely loss of function. And the mechanism can be, altering the open probability, altering the conductance, or decrease the availability by trafficking defective mechanism, which largely would be the case for ion channel because they're membrane proteins. It needs to get to the cell surface of your cardiac myocytes or your neuronal cells. And the other way it can cause trouble if you have a variant that is, say, affecting the splicing or near the exon intron boundary, it would then, would not have the mRNA, would cause the haploinsufficient. In this case, it would decrease the number of availability of the ion channels at the cell surface. All this will then lead to reductions in your current, which is what you will see, or we can measure using the patch clamp electrophysiology by quantifying. In this case, the example is loss of function. So in the cardiac channelopathy space, so I have an action potential here. You can see we have the sodium one inward current, and then the calcium and the potassium outward current here. And all the different action potential in the heart comes together to give you this ECG. And the first bit would be depolarization. The second bit would be repolarization placed by different ion channels. So the first one I'm going to talk about is the sodium channels. So this is the wild type, and if you have a variance that affect trafficking, say, then you have loss of function and you have little current to measure. But in the SCN5A or 1.5 sodium channel, you can have gain of function that cause long QT3. If loss of function is Brugada, gain of function is long QT3, where you have slow inactive or late current here, that would then prolong the action potential, will prolong your QT here, which is why it's called long QT. And the other one is then the calcium channel, which the wild type here, you have a bit of late current normally, but then if you have a pathogenic variant, in this case, it's the Timothy syndrome G46R variant, and it can cause Timothy syndromes of the long QT depending on the clinical presentation of the patient. And now it comes to potassium channel. So we have two potassium channels here. One is conducting the IKS, the slow delay rectifier, and it comes together with a KCNE1 to form the KB7.1 long QT1, and it can also cause the JLN syndrome. And this is a typical wild type slow IKS current. And if you have a pathogenic variant that causes trafficking defect, and you basically have no current that we can measure if it's a severe loss of function variant. The other one is another outward potassium current known as IKR or KCNS2 that can cause long QT2 if loss of function in this gene. So we measure the outward current here for the wild type, and you can see the pathogenic one typically have nothing to measure if it has a large effect size. So today I'll start off with KCNS2 because it's one of the genes that we have the most data for, and it's where we set up initially to see whether it's actually feasible to use the automated patch clamp to study channelopathies in the clinical setting purposes. So the way we design our experiment is, in this case, because it's potassium channel, so we co-express the variant with the wild type because potassium channels are different to sodium and calcium channel, where you have 4 identical subunits comes together. So if you have a mutation, in theory, you can have 4 subunits have a same 4 mutations, or you can have 4 wild type or 50-50. So it is a combination compared to if you have sodium or calcium channel, you have 1 mutation per functional channel, but potassium channel can have a mixture. So to investigate or capture the dominant negative effect of potassium channel variants, we have to co-express with the wild type. And all these are then generated into stable cell lines, and we typically assay 10 variants plus the wild type and negative control on a single patch plate that Will had described to you. And we, arbitrarily speaking, you can have it anyway. We have 12 lines on each plate. So we typically have a wild type, negative, and then 10 variants in the middle here. And each of these little square here is a single patch clamp experiment. So you can have up to 384, but it doesn't mean you're going to get 384 good recording. Typically, with all these experiments, we apply very stringent quality controls. Above 50%, it would be a good day, I think, because we perform a technical replicate. So if you have a 50% technical replicate, you get back your 384 well, I suppose. And we also then, bring up the cells again on a different day, perform the experiment again. The order would be different, would be randomly across. It wouldn't be the same order as we did the first time. The reason is we wanted to be able to reproduce the data, even though it doesn't matter when it was performed, and the results should be similar. The mean values we get for the variance should be similar after normalizations to the wild type from the same plate to control the plate-to-plate variation. This is because we produce functional data directly for the clinical testing laboratory. So we want to make sure what we produce are accurate and consistent, because it will affect the patients if we get it wrong. So these are some of the things that we consider when we look at the QC and whatnot for our result, because it's a voltage-gated ion channel. So, voltage and current are sort of related. And I have a ventricular myocyte action potential example here just to show you for IKR, which is passed by KV 11.1 here. You can see IKR really coming out during the repolarization, and this is what the channel is for, to repolarize your cardiomyocyte back to the resting state to maintain then the long normal QT interval. So if you lose the IKR, then of course your action potential prolonged, then your QT is going to prolong. But, as you can see here, the peak IKR is about, in this case, estimated to be -50, which is why we design our voltage protocol. We open and activates all the hub channels or KV 11.1 channels, and then assay the amplitude, which is represent the functions of the channel at -50 to represent, the regions that the functions of the channel that is important. Because biophysically, KCNH2 or the hub potassium channel actually pass more current in the inward. At, say, for example, if you use a -120, the current amplitude is actually a lot bigger than the outward current, but in the heart, it's not going to do that. So even though biophysically you can interrogate it that way, but we want to capture and measure the function that is physiologically relevant in the artificial hectonitrile cell system to the best that we can. So the next thing we do, then we can quantify a lot of this wild type current density to produce this plot. And then you can see it's not normal, not Gaussianly distributed. We can transform using a square root because we are using a Z-score system that I will show you later. So we wanted to know the mean because we need the mean average values. And the other way this is done is to then allow us to estimate the smallest standard deviation that we can achieve by measuring, in this case, 1,000 cells. And with that standard deviation, we can then determine minimum sample n number that require to be able to consistently produce the mean values that will be consistent across different experiments. Because once you reach that critical n number, typically the mean value doesn't really change that much. It will fluctuate a little bit, but it will not be drastically different. So which is important for us. And with sodium channel recording, we also want to make sure we actually nowadays use holding potential at -90 to measure the peak current at -30 to the same reason that we design for the potassium channel. So we don't hold it -120 for the sodium channel recording when we measure the peak current, because then actually more channels will be available than it would be in the heart if you're holding a more negative potential. And for quality control, because sodium channels are more, I guess, sensitive to the voltage or quality of the clamp, as in the applied voltage to the cells. And if you have a large current, sometimes you get plot like this, and we want to make sure everything that we analyze will automatically QC out, bad recording. Only good recording will be proceed to summary without, subjectively excluding data. So we engineer everything in a QC automated pipeline. So the first KCNH2 patch clamp assay was published in 2020. And at that time, we just simply wanted to ask the question if, feasible to use a patch clamp or APC system to discriminate a pathogenic variants from benign variant in this gene KCNH2 that code her potassium channel or KB11.1. But at that time, we published the paper, and afterwards, the ClinGen recommendation published the recommendations for how to apply the functional evidence using the PS3 and BS3 code, and they listed a series of criteria required for, I guess, clinical applications or functional data. And of course, our results or our assays would not be satisfying this recommendation at that time, because we don't really know what the recommendation was, because it hasn't published them. So which is the reason then I follow up the assay with collaboration with different testing laboratories and to develop and calibrate the assay that the clinical lab will be happy to use. And in the next few slides, I'll talk about how we do it, so that we can now apply the KCNH2 assay in a clinical setting. So the idea is really, according to the Greenwich ClinGen recommendation, is you need to have large number of variant control. And sometimes you can't get large number of benign controls in ClinVar because that depends on how it was classified and whether they classify those common variant or not. And typically, what we end up doing these days is we go to NOMAD and then select the high allele frequency variant that the frequency is greater than the prevalence of the disease, which cannot be the cause of causing the disease in those patients. So those will be using as a healthy controls. And in this case, we have 30. And simultaneously, we then curated 30 non-pathogenic variant, but we don't simply go with ClinVar and take those. We actually use internal database from those testing laboratories, curate large number of proband and whatnot, and then ask clinical geneticists to actually independently assess them again in the study. So we make sure everything is done correctly. And after all these are curated, I generated them into stable cell line, measure the current. So this I have described before. We measure simply the peak current in this case, because more than 90% of the KCNH2 variant are causing trafficking defective phenotype, which then reduce this peak current. And the benign controls here from the NOMAD largely would be having similar current density, fluctuating more or less in a way. And then the disease-causing variant would be mostly, if a large effect size, you have nothing to measure, but sometimes it can be partial loss of function as well. So here I start off with the benign controls, the 30 benign controls. As you can see here, this is a range from the highest to the lowest. And to allow us to then generate or determine the Z-score system that we use, in this case, when we interpret a VUS in the future. And the Z-score system is simply you're taking the mean of all the 30 benign controls here and 2 standard deviation within the mean of the population. 30 benign population variant here would then be determined or defined as functionally normal range. And from this assay, so this is just to say that we, can capture the biological biological variation from the variant that exists in the population, so that when we call something that is abnormal function, it would be very likely to be abnormal. So anything below -2 would be then considered having abnormal function. So this would be capturing 95% as opposed, full standard deviation. So then, as you can see, the 30 pathogenic controls here, most of them have large effect size. So, you're smaller, full standard deviation away from the population mean. And some of them are partial loss of function because it's a continuum. You don't suddenly just drop off. But the cutoff here, because of that, we actually use greater evidence strength, even though the assay itself, if you count, how the sensitivity and specificity of it can be applied at PS3, but we use a greater evidence strength. The yellow region here between -2 and -3, we use as supporting. moderate, and then severe large effect size, we use a strong evidence strength using the PS3 criteria. So this assay now satisfies the cleansing, and we have used it in a few case studies. So using our Z-score system, we measure the variant that being referred to us. If there's a large effect size, then you have a very small Z-score, and then we'll be able to apply the PS3 as strong in this case for these guys, and you would then be able to reclassify the variant most likely to at least likely pathogenic, because if you remember, PS3, the strong evidence were 4 points, and you need 6 points to be a likely path. You can combine with other evidence strength. And this is also functional data alone will never be sufficient to classify a variant as likely pathogenic. What we determine is the variant is abnormal. And similarly with this variant here, you can see one of them is severe, large effect size, but the other one is normal. But the one that is normal actually affects the splicing. So it wasn't because of not having the current density. You can look the paper up if you're interested. But those are some case studies that we do. But to test the assay in a clinical setting, I collaborated with Invitae. In this case, Invitae used Sherlock algorithm to classify variant. It needs 4 points to reach a likely pathogenic. And from Invitae's VUS, I have 193 of them have a patch clamp data set available. And of the 89 VUS, Invitae can apply 2 pathogenic points using their Sherlock. This means it's 50% day to reach the likely path. And out of these 89, 26 be able to be upgraded to likely path of pathogenic, affecting 66 patients. But the other 63 VUS, even though they have pathogenic points here, meaning they're abnormal, they still be a VUS at this stage because, again, you need other criteria to come, to a likely path in this case. But they would be already moved towards likely path in the future when more evidence are available. So the other project that is ongoing is to work together with the UK laboratory. So Kat Thompson here is the principal geneticist from the Oxford testing lab. So we come together to reassess all the KCNH2 variant in the missense and India from the NHS testing laboratory to see, in this case, using the standard ACMG AMP criteria at the national level, how useful it is actually the functional data. So in addition to what I show you, enhancing genetic diagnosis, can we predict risk using functional data is the next thing that I'll talk about. And in this project, I have generated 533 missense variants from long QT2 cohort around the world. And here I just show the podomain variant first. And the blue region is the same normal function range that I determined before using the Z-score system. And the blue variants here mean they are severe loss of function. And this is not new because we know already published by Led Arthur 23 years ago, if you have a poor variant in KCNH2, you're gonna have a higher risk and if compared to the N or C terminus. But now if we look at the N terminus variant here, which is considered to be passed to here, you can have a mixed phenotype. If it's in the structural region, it depends on your amino acid changes. It can be severe or normal in terms of what we measure by patch clamp. But if it's in the unstructural region, most of them we found are normal function. Similarly, it is also true for the C terminus. If you in a structural region, depending on what the amino acid changes, but most of them will be bad. But in the unstructural region, mostly would be normal. And here you have a lot of VUS because they are rare variant according to Norman, I suppose. But a lot of them are normal function. So this is sort of correlate pretty well with this what Arthur has shown previously, but we now actually have a variant specific data to perform a risk prediction. And these are just the cohort that we have. So Brett here did a modeling of the 20-year series event by using just the patch clamp, the sex, and the QTC. And there's a MAVE data set for the KCNH2 where almost, all the single nucleotide variant will be available for the KCNH2, but this is a patch clamp talk, so I'm just going to focus on the patch clamp data here. At a 25% difference, we already see it has a predictive value, similar to the QT changes. But if we use APC, a 50% change, which is your haploinsufficient scenario, which we know causes long QT2 syndrome, it's actually is very useful to predict, if you have that variant, what is your risk compared to if you have a variant that has a normal function. So we definitely can tell if, a patient that is referred to as for functional testing, we can tell whether that variant has a high risk or moderate or low risk. So that is for KCNH2. So the rest of the talk, I'm going to talk about SCN5A and NAV1.5 causing Brugada syndrome. And this is a collaboration with Andrew from Vandenberg. And again, Brugada syndrome is a loss of function, which we can confidently measure using patch clamp that I described before. The experimental design is similar, where the geneticist or genetic counselor curated the benign and the pathogenic controls. We then made the cell lines and then generate the data. As you can see, we can segregate the benign from the pathogenic except for one of them. So we don't always get it right, but the performance will still be high enough to allow us to apply a PS3 strong, again, for the large effect size variant, moderate and supporting, depending on what the Z-score the variant is determined. So then we wanted to test how useful it would be in a large data set. So Roddy was published this back in 2021, where there is about 252 missense and indel SCN5A variant in the Brugada cohort. So we assay together with Andrew. So the co-first author, Matt from Vandenberg and Joanne from Victor Chang, undertake these studies. And this is just to show that this is the same benign and pathogenic controls I showed you in the previous slide, and these are all the patient variants. So we found that a large proportion, roughly in this case half and half, say, have normal and abnormal function, and you can have wild-type like function, partial and severe loss of function. And remember, these are detected from patient, but for Brugada syndrome, it's a bit tricky, I suppose, because we don't really know sometimes, the SCN5A is really the cause or not, but it tends to believe if you have a SCN5A variant detected in the Brugada patient, it, most likely will be, but we found, 40% or 50% is having a normal function in our assay. And if you then plot all the location of this variant out on a topology, you can see, most of them, the severe loss of function are in the codomain region, which makes sense with the biophysical property of the protein. And the normal function variant is in the interdomain linker, which is the unstructured region, I suppose. And it makes sense and consistent with the protein structure. But whether it makes sense clinically in terms of, whether it can cause the Brugada or not in those patients that have the variant. So then Andrew did a modeling by simply taking the Z-score, which is the functional measurement that we have, and then calculate the penetrance of the Brugada by looking into the population NOMAD database to see, if you have a normal function compared to the one that you have severe loss of function, large effect size, and you can see the large effect size will have a higher penetrance, similar to what the Brugada syndrome is in the population. And this sort of, suggests that what we measure using the patch clamp, sort of consistent with the clinical observation. And to then see how useful it would be, regardless of whether this is true or not, for genetic classification with the evidence level that we determine. And this is pre-functional, meaning we haven't applied functional data. You can see most of them are VUS, and this is already applying the ClinGene calibrated in silico alpha missense in silico score, but still a lot of them are still VUS. And if we then apply what we determine as different evidence strength of the functional evidence, we can see if you have a strong functional evidence or moderate, you will likely be able to reclassify to likely pathogenic in most cases. And if you are supporting some, half and half, you still, some of them will be a VUS, some of them will be likely pathogenic. And if you're normal function, of course, in this case, it will be either VUS or likely benign. We apply a very stringent criteria to reach likely benign, which is why a lot of them are not. And this work is currently under review, but medRxiv paper is available. There are a lot more data in the medRxiv. If you like to go through it, you can go and check it out. So in addition to KCNH2 and SCN5A, and we also do KCNQ1, E1 now, and as well as the L-type calcium channel for the cardiac channelopathies. But in the lab, we also do other channelopathies related to brain, muscles, calcium channel. This is collaboration with David Adams, and the potassium channel, the KCNQ2 and Q3. And these are all currently happening. The idea is to provide patch clamp data for the patient that needs evidence for reclassifications in Australia and overseas if I get contacted. So lastly, just to finish the talk by acknowledging the team, my collaborator and my genetic collaborations and industry and the author of the KCNH2 and the SCN5A, and happy to take any questions.
Thank you very much, Chang. There's such an outstanding body of work that you guys are doing. The Q&A questions are open, so if anyone in the audience has questions, please feel free to write something in the box, and then we'll get those answered. To give us all some thinking time and breathing time, I'll start us off, Chang. How long does this process take now from when a patient presents to the clinic with some form of cardiac anomaly? How long does this process take for these cell lines to be developed, for the ethers characterization to happen, and then for the stratification of risk these days?
Yep. So I work closely these days with the Sydney Children's Hospital in Sydney. So I got referred a variant today again for Brugada syndrome. And from ordering the plasmid to produce the data, it's generally about 3 months. We can generate the cell lines, stable cell lines within a month upon receiving the plasmid. The plasmid probably take 3 weeks, depending on how large is the order. If you order small number, they come quickly. But if you order 50 variant, they might take a while because, that's the way they have to repeat some of them sometimes. So yeah, 3 months we can generate a research report back to them, and then we then liaise with the testing laboratory to then go through reclassification. Yeah, 3 months is what we.
Fantastic. Thank you very much. We've got some questions coming in now. First one from Jerome Montnach. He says, "Thanks, Andy, for an amazing talk. Have you correlated the APC severity of a variant with clinical QT enlargement?"
So in one of the paper that I showed, the AJ actually is the 2024, forgot the journal, HGG Advance, I think, where I calibrated with Ken Thompson. In that paper, we have a pediatric cohort of non-QT2 from Germany, Munich Heart Center, I think. And we correlate the QT that have measured to the variant function that we measure from the patch clamp. And there is a correlation there in that small cohort. So if you can, I didn't show the figures, but if you check that paper out, you can see it's largely correlated. If a large effect size loss of function measured by patch clamp, you tend to get prolonged QT compared to if you have a normal function variant.
Right. Oh, nice. It's a good sign, at least, if there is some correlation there. It's good to hear. A question from our own Rodolfo from Nanian Inc. in the US. Do you envision the SynchroPatch becoming a complementary diagnostic tool in the clinic?
I will say no, because for the reason it's quite expensive to produce the data. in a way. It depends. I suppose if you are a well-funded diagnostic lab, then of course it can be, but otherwise it would be in a research setting typically because I just can't see the normal clinical testing services, acquire and operate a single patch in their pipeline. Yeah.
Yeah, I can imagine the need for trained personnel might be tricky as well to fit into that pipeline.
So which is why we try to work with them. And then I suppose this can then answer your question whether it can be a complementary diagnostic tool in the clinic, but not operated by the testing laboratory itself. But we, in the research, generated the data to allow them to then use the data in a way, I suppose, is still a complementary diagnostic tool in the clinic. But it's not easy because at the beginning, we face a lot of hurdle from the industry or the testing labs in Australia, at least, because traditionally they are very conservative in applying functional data at a higher than supporting level, which then means the functional data, the utility is not great if you just apply a supporting, even though that variant is severely loss of function that you can determine functionally, but they will not apply at a higher than supporting. So we had gone through a series of iterations, educating and presenting and show how we calibrate the assay. At least in Australia now, they are more comfortable using it, at least moderate if it's large effect size, if not as strong. So again, it depends on the testing laboratory, how comfortable they are. So this will also then contribute to conflicting classifications in ClinVar, depending on the laboratory. Say if one laboratory apply a functional evidence at strong level, the other apply as supporting, the rest of the criteria remain the same, then you might get a VUS in one testing lab, you might get a likely path in another testing lab. So that becomes a problem of your genetic test result. Depends on where you send your result to get your test at the first place, which is unfortunate.
I see. Another question here from Jerome Montnark. For the remaining VUS variants, have you investigated other parameters other than amplitude to explain pathogenicity?
So for KCNH2, we did investigate the rate of deactivation as well to see whether that correlates. But a lot of the variant will already have trafficking defective phenotypes rather than it can also have rate of deactivation problem, as in the channel would close faster to reduce your protective current, I suppose, during the different systolic, I suppose. But because when we calibrate the assay, we need a control variance. So we need to have a big number of control variants that will have a rate of deactivation problem to be able to then confidently to see that effect. So in that sense, we only applying supporting evidence if we find something that is affecting the rate of deactivation, but mostly already captured by loss of current amplitude in KCNH2. But for SCN5A, as in NAV 1.5, for Brugada, again, it's loss of function. By measuring the current amplitude, we found the other parameter is not useful, but for gain of function, other parameters would be very useful, which you will have to use to then tease out all the different abnormality.
Now that makes sense. A question here from Stefan Harmer. This is a very nice talk, thank you. Regarding the heterozygous KCNH2 variant APC analysis, have you tested if placing the variant after the IRES rather than before, as in your flip-in design, makes a difference? He asks because he's interested in the importance of modeling the heterozygous state.
Yes, it does make a difference. So we actually make a 1A, 1B line using the IRS. So we place the 1A IRS 1B or 1B IRS 1A in that way. So 1B is another isoform of KCNH2 where you have a fast DX. So you will be able to see that if you have them co-expressed together. So we found we need to have a 1B in front of the iris and then followed by 1A. So meaning the 1A would be less in that way. So which is why we design our variant. We accept the limitation. We accept that having some wild type is better than no wild type, even though it's not perfect 50-50. So we design our variant first and then iris and then the wild type in a way in our bicistronic vector design. But we in the lab now actually now making double flipping where it's actually two plasmid together separately come together to express different allele, and we'll compare that to how different it is by testing a few variants to see.
Right, I see. Question here from Mark Rogers. He says that Al George is doing similar channelopathy profiling with APC and has noted some issues with genomic stability of NAV 1.2 clones in cell lines. Have you seen this or checked for the sequence accuracy with your NAV work?
We get all our sequences from Al George. So he worked it all out and then we use
Nice.
what they found. So hopefully we won't have the same issue.
Easy, great. A question here, why is 50 milliseconds QT length is selected for comparison with APC data prediction risk in long QT2? This is regarding the O'Neill paper 2024.
So that will be a question that I probably can't answer you correctly because Brett is the one that did that modeling. Whether that is 50 millisecond prolonged from, I can't remember exactly, is it 50 millisecond prolonged from 460 maybe from the baseline to reach the 500 threshold?
Okay, no, that's fair enough. I've only got time, unfortunately, for one last question. We're a little bit over time already. And that question here would be, in what sense does the level of mutant channel transgene expression affect the patch clamp outcome?
You mean, I guess the question is the current density?
Yes.
As a problem. For sodium channel, it's a problem because if you have a large current, then you can patch it. Oh, you can vortex clamp it accurately for your measurement. So then you might have to adjust your solution, your sodium concentration, depending on your current expression. Because we use inducible line in our system, so we can actually also adjust the induction time of doxycycline. We normally do 48 hours of induction, but if it's too big of a current, we might, do a shorter induction if required. But this we generally don't because we just do 48 for consistency. Just want to reach the steady state for the wild type and different variant. Yeah.
Okay, great. Chang, thank you very, very much for this. I think I will close the Q&A session there. We're a little bit over time. For those people who have not had their questions answered, please feel free to get in touch with us or with Chang, and we'll certainly get back to you with those answers. Chang, thank you very, very much for joining us today and for sharing your outstanding work on the syncropatch and with these channelopathies. It's really inspiring to see how people are actively being helped by this kind of work and being able to reclassify these VUSs. That was really fascinating to see. So thank you very much.
Thanks for having me, and I hope everyone have a great day.
Thank you, and same to you, or at least a good night as well in Australia.